Hello. So what I want to do today is give you an example. Sorry, let's have some water first. To give you an example of a technology development, a quite recent one, that seems quite promising in terms of translational perspective and could be of interest to some of you. So to set the stage a bit, my lab is interested in molecular processes of tumor evolution. So very general things, it has been clearly shown now for, quite some time, that in terms of molecular evolution processes, there is also a contribution of both genetic and non-genetic processes. And what's really interesting, and is a lot of interest to us, is that you can see at the same time selection of rare pre-existing features, as well as acquisition of novel molecular traits. And this is really important if you think about the translational perspective of this, because it's not the same to treat things that already exist and target them, and to target things that are acquired along the way during treatment, for example. So what I'm showing here is actually two of my favorite latest examples of really how a single cell has been able to shed light on mechanism of tumor evolution processes, and really in the context of cancer therapy and resistance to chemotherapy. And so, of course, it has been now clear that single-cell techniques are revolutionizing the way we think and we appreciate tumor evolution processes. And here's the example of DNA and RNA sequencing telling us, again, that there's both genetic and non-genetic features that are going to be already there, but also acquired for during the treatment, and here, chemotherapy. In this case, again, it's most on the microscopy side, but this paper is telling us that there are transcriptomic features that are already there in the cell population, but you need to acquire additional features in these cancer cells to become fully resistant. So here it's an example of DNA and transcriptome, and I'm here to talk about chromatin. And this is really the early days of single-cell epigenomics. Most of you already know that. So what we're trying to do in the lab is really to tackle the concept of chromatin plasticity. And if I may just bring about this concept, it's clearly shown now in cancer that we have an abnormal really distribution of chromatin state. And the field is really getting now at the moment where it's trying to understand why and how this is happening, what is giving to the cancer cell. And what we think, and many lab thinks, is that those chromatin landscapes are really setting the stage for what transcriptome can do and cannot do. If you think about repressive chromatin states like H3K27 through methylmark that are here to repress transcription, well, it's believed that displacing it around is going to tell you either if you repress abnormally chromatin state, you're going to really prevent transcription from happening. But in cancer cell, if you lose it, you might be giving the potentiality to the cancer cell to have transcription happening at these loci where you're losing this mark. So what I'm telling you is that having this chromatin landscape information is completely complementary to knowing what's happening at the transcription level. It's not equivalent. Chromatin is a potential, and the transcriptome is telling you what's really happening. So when you talk to MDs, I'm really trying to tell them that we need this chromatin information to have a complementary view of what's happening in terms of molecular processes. And so our aim here is to understand, in my lab, the heterogeneity of this chromatin feature and really have the notion of dynamics in time in tumor evolution processes. So this is real nice, but when I started, we needed technologies to do that. So that's where we teamed up. Sorry. Yeah. I don't know. The video's, yeah, it's going to work, maybe. So that's where we teamed up with physicists, microfluidics expert, and we set up really a workflow to study chromatin marks, so chip, at the single cell level. And just to cut a long story short, this is really, if you compare it to single-cell transcriptomic, for example, where you have one step of microfluidics, here you have three. So it's really thanks to the expertise of microfluidics that we manage to really have chromatin prepared from single cells. And these chromatin fragments have a barcode like we see in every traditional single-cell experiment. And so when you perform your IP, you can guess and you can really read in the data which chromatin fragment is coming from which cell, and etc. So with this type of pipeline, we're now able, at Institut Curie and in other places at the SPCI, able to perform single-cell chromatin profiling of many different marks on 3,000 cells and really have an idea of how these marks are distributed in terms of in complex samples as tumors. So this is just to tell you, for those aficionados of single cell, this is how the data looks like. You have about 10,000 loci that you can really immunoprecipitate per cell, so it gives you quite a thorough epigenomic map of each cell. And this is the reality, okay? Everybody tell me, what is the data? Each line here is a cell. And what you see is that the data is quite sparse, okay? But it's really getting there. We're getting to the point where we can see, for single cell, like H3K27 three-dimensional distribution in this case. And what's really nice with this kind of data is that we're working on a droplet system. We don't have those many batch effects as we do see on the 10x kind of platforms. So what did we do with it? The first thing we did was to profile a PDX, or patient-derived xenograft system. So we were fortunate enough that a patient from Institut Curie gave her luminal tumor for research. And so we had one part of this tumor that stays sensitive to tamoxifen and the other that became resistant after several rounds of tamoxifen treatment. In this case of resistance, or acquired resistance, we didn't see any copy number alteration or major driver of resistance that were mutated. And what we did see, we profiled, so we really led, without any a priori, a data-driven approach. And when we profiled at the single-cell level chromatin of these different tumors, what we saw here, so let me walk you through it here, it's all the cells versus all the cells. And the color is telling you how much the chromatin landscape, H3K27, is correlated between one cell and the other, genome-wide. And what you directly see is that if you mix the cells from the two models together, you see that there are two main epigenomic profiles. That's easy to see. But then what you also directly see is that some cells that do come from the sensitive tumors are highly resembling, have the same epigenomic landscape as the one coming from the resistant tumor. And these cells make about around 16% of the cells. If you challenge the data, you remove some cells, remove some loci, it doesn't move a notch. There are still 16% of the cells from the sensitive tumor that look like the resistant cells. So we were quite surprised to see this was invisible at the bulk level. We couldn't detect this population. So just to tell you that the data now is good enough to perform, so as you've seen, clustering and without any a priori, but also to perform differential analysis at the single cell level. We can pinpoint the loci that are really characteristic of these different epigenomic layouts. And what we saw actually is that a lot of things are moving around. Mostly we're seeing demethylation in the resistant and resistant-like cells, which was kind of expected actually. And it's happening at loci that do make sense. So you maybe cannot read here, but it's happening at loci that are involved in mammary cell identity. So what we're thinking here is that perhaps we're seeing demethylation at genes that are involved in changing the identity of the cells from luminal to basal, for example. So it's really, we're really now studying this differential enrichment loci. So just to finish on that, you can tell me, well, what would you have learned doing the standard 10x transcriptomic analysis? And this is where it got interesting. More interesting, I would say. But what we did actually was on the same pair of tumors. Forget about what I said on the epigenome. We studied also with a standard single-cell RNA-seq approach to say, well, what can we learn at the transcriptomic level of this pair? And actually what we learned is that there's a huge heterogeneity in either the starting tumor as well as the resistant one. And what we learned is that one of the cluster here, the red cells, well, they already activate a lot of the pathways that are activated in the resistant tumors. And you find the same kind of guys that we saw before, the ones involved in basal breast cancer genes, but we also found some additional pathways, saying that, again, and this is 17%, at the transcriptomic level, there's also a group of cells that do show non-genetic features of resistant tumors. And it's more features than what we saw at H3K27, but it kind of makes sense because H3K27 is not responsible for the entire life of regulation of the cell, right? So it was completely independent. So just to tell you, so we're actively pursuing that, going for tumor samples and so on. But I think it's really important to really see that a strong technological effort that was really a collaborative achievement between 3 labs was really key to having this high-tech and a good data quality for this single-cell chromatin profiling. And that's now really enabling a lot of data analysis. We found, so by different means, that there was this subpopulation of cells with non-genetic features that were already present. So really, again, tackling what we thought about epigenetic plasticity. Well, maybe not. Maybe some epigenetic features are selected for just as mutations. So this is clearly something we're studying right now. And just, I want to finish on this very simple scheme, just trying to say that this is all the changes that we're going to see between 2 states and 2 like resistant and sensitive one. What we're tackling with single-cell RNA-seq might be really the top of the iceberg. The things are already highly expressed and moving around. But what I'm saying is that we need other methods, like the single-cell chip, for example, is maybe tackling things that are independent of any expression level, for sure, but they're only a part of what we're seeing in terms of expression differences. Just to say that we need those orthogonal methods, seeing RNA-seq is not all, seeing R-Taq-seq is not all. We need to really tackle these kind of molecular evolution processes by different means, and data integration here is going to be instrumental in the sense that we're not seeing the same things with those different data. So how do you integrate when you don't interrogate exactly the same space? I think it's an important question. And for sure, what we're doing right now is moving to two patient samples on this. So I'll thank you for your attention, and I'll take any questions.
Thank you very much for a beautiful presentation. Yes, we have time for one or maximum two quick questions. Yes.
So, thanks. Do you think that the chromatin-based identities are stable over time or plastic?
So, well, I think it depends on the mark first to start with. In case of H3K27, that's why we chose it, because it's kind of stably transmitted through the generation, but it's still plastic because we know in development that it needs to change around to really define the cell identity. So what we're doing right now is to combine this to linear tracing approach, for sure, to test really if it's the same cells that are sharing their pattern, or there's a kind of stochasticity that's being selected for. So that's what I cannot really answer right now. Yeah.
One very small question. So the problem with epigenetics is, we are doing it also, is that you have many marks. So where do you choose from, and how many do you need to assess to get actually the true capacity of the cells to react?
So yeah, so I think it's a very important question. For Taxic, for example, because people always ask the question, it's 1% of the genome that's interrogated. These open regions make for 1%. Here we have tried to take the approach to look at repressive mark. We make about 10% of the genome, but we need to reach 100%, right? So I think it was detailed in the ENCODE project. I need 5 marks to make 100%, so let's achieve 5 marks. It would be already a good thing, right?
Okay, thank you very much.